Mesdames, Messieurs, chers collègues, alors tous mes voeux à tous pour cette année nouvelle. J'ai le très grand plaisir et le très grand honneur d'accueillir aujourd'hui le professeur Elaine Fuchs dans le cadre de sa nomination comme professeur invité au Collège de France. Je n'ai pas regardé si c'était la première femme ainsi nommée, mais si ce n'est pas la première, les doigts d'une main devraient suffire. Longue ou courte introduction de la carrière d'Elaine Fuchs, je n'ai pas eu longtemps à m'interroger, l'attaque d'un virus à tropisme pour les cordes vocales a fait la décision. Cependant, en quelques mots, Elaine Fuchs est chimiste de formation. Elle a fait une thèse consacrée à l'étude biochimique des peptidoglycanes bactériens à l'université de Princeton sous la direction de Charles Gilvar. Elle a rejoint ensuite le laboratoire d'Howard Green au Massachusetts Institute for Technology pour un stage postdoctoral. C'est là que s'enracine le début de ses travaux scientifiques, depuis lors consacrés à l'étude de la peau. Ce laboratoire avait mis au point une remarquable technique de culture de la peau. À partir de biopsies, de grandes surfaces de peau pouvaient être obtenues dans des boîtes de pétri qui ont bien vite prouvé leur utilité pour les grands brûlés. Elaine Fuchs va ensuite s'intéresser aux kératines qui composent les filaments intermédiaires des kératinocytes et montrer que l'expression des gènes correspondants se modifie au cours du développement et de la différenciation de la peau, sujet d'étude qu'elle poursuivra ensuite à l'Université de Chicago. Elle réussira à attribuer une maladie héréditaire de la peau, dite épidermolyse bulleuse, à l'atteinte de gènes qui codent des kératines K5 ou K14. La peau, enveloppe protectrice, soumise à des agressions microbiennes et des tensions mécaniques, est le siège d'un renouvellement permanent. Grâce à l'initiative d'Elaine Fuchs et aux possibilités d'étudier cette peau chez les animaux génétiquement modifiés in vivo et en culture, cette peau, devenir un modèle de choix pour comprendre la biologie des jonctions intercellulaires, comment se défont et se reforment ces jonctions, quel est le rôle du cytosquelette. Elaine Fuchs va non seulement en pionnière, mais presque en solitaire décrypter bien des aspects de la biologie moléculaire des kératinocytes et porter à un degré d'analyse inégalé jusque-là la définition des événements cellulaires et moléculaires qui sous-tendent la formation des jonctions cellulaires. La peau se renouvelle, ses poils en particulier. Elaine Fuchs, qui dirige un laboratoire au Rockefeller, qui appartient au Howard Hughes Medical Institute, est maintenant engagée depuis plusieurs années dans l'étude de l'homéostasie de ce tissu, c'est-à-dire dans la recherche d'une vision intégrée des équilibres cellulaires dynamiques des populations qui le composent. Elle approche ce problème par une étude hautement résolutive de l'origine des diverses populations cellulaires qui donnent naissance aux follicules pileux. C'est ainsi que depuis plusieurs années, Elaine Fuchs se consacre à l'étude des cellules multipotentes de la peau. C'est de ce sujet dont elle nous parlera lors de son second cours la semaine prochaine. Elaine Fuchs, par sa connaissance des jonctions cellulaires, de la polarité cellulaire et des cellules souches, se trouve naturellement au cœur des concepts développés aujourd'hui à propos de la transition, auto-renouvellement des cellules souches et processus de cancérisation. C'est ce qu'elle discutera lors du troisième cours. Aujourd'hui, elle va introduire cette série de cours par une présentation générale sur les cellules souches. Les avancées scientifiques réalisées par Elaine Fuchs jouissent d'une très grande reconnaissance internationale. Elle est membre de l'Académie des sciences américaines depuis longtemps. Elle a reçu de très nombreuses distinctions internationales, dont le prix Lansbury que nous connaissons bien ici, puisqu'il est délivré conjointement par l'Académie des sciences américaines et françaises. Nous nous réjouissons de sa présence parmi nous pour les 3 mois à venir. Et maintenant, Elaine, tu as la parole.
Je vous remercie, cher professeur Petit, pour votre présentation chaleureuse et écouillante. C'est un grand honneur de vous parler aujourd'hui. Dans l'atmosphère, cette salle de conférence, j'entends les voix des nombreux chercheurs et savants qui ont donné des conférences depuis le fondement du Collège du XVIe siècle. À l'ampleur et à la beauté de ce bâtiment, je sens le dévouement historique de la France en faveur du développement de toutes les sciences naturelles et humaines. Malheureusement, il me manque le style des poètes de la Pléiade qui pratiquaient leur art pas loin d'ici. Enfin, je ne parle suffisamment bien le français pour vous présenter ma recherche dans votre langue. Par conséquent, je suis contrainte d'utiliser l'anglais, la langue internationale des sciences aujourd'hui. Mais je me souviendrai à jamais de l'occasion qui m'a été donnée de faire cette conférence en un lieu unique et reconnu pour ce qui concerne la liberté d'esprit et l'imagination scientifique. Je vous remercie encore, professeur Petit, et vos collègues pour cette invitation. And now my lecture will be in English. I thank you for your patience. So I thought that for my first lecture, since I'll be talking about stem cells, and particularly the work that my laboratory has been doing at the Rockefeller University, that I begin with an overall view of stem cells, their biology and their promise for regenerative medicine. And so today's lecture is going to be very general, and yet at the same time, for those of you who have no scientific background, I'm hoping that you'll capture something from this lecture. And for those of you who have a very strong scientific background, even one in stem cells, I'm hoping that you'll be able to capture something from this lecture. So I'll try to provide you a spectrum, and then on next Tuesday to focus more on the particular research that my own laboratory does. So at the Rockefeller University, my laboratories are located here. They're on the top floor of this precarious building positioned right over the FDR Drive in the East River, looking straight down on the United Nations, which I see every day from my window, and then down to the Brooklyn Bridge. This is the Rockefeller campus, and we're right in the heart of Manhattan. So on the 4th of July, my laboratory sees this view outside of my window and my laboratories, my postdocs and graduate students' windows. During the rest of the year, we have to generate our own fireworks, which we try to do indoors, and we do so by virtue of trying to address interesting and exciting questions. One of the general questions that we're interested in is that of stem cells, and just so that we're all understanding this area, stem cells are cells that are able to make and replenish one or more tissues. And they typically exist as relatively slow cycling or infrequently cycling cells that then can give rise to transit amplifying cells, cells with a limited number of cell divisions, before these cells go on to differentiate to make particular tissues. In the case that my laboratory works on, this would be the epidermis, the hair follicles, and the sebaceous gland. But the paradigm for stem cells is one that applies to many different types of stem cells of the body. Stem cells have the unique capacity in that they can divide symmetrically to generate self, so that stem cells can self-renew, and in addition to that, they can divide asymmetrically in order to undergo this differentiation to make specialized cells that constitute the tissues. So the difference between an embryonic stem cell and an adult stem cell is really one of plasticity, or the options that are afforded to the stem cell. The embryonic stem cells are pluripotent. These are cells that can generate all of the cells of the body, and the only cells that these embryonic stem cells cannot make are basically the support cells for the fetus, the placenta and the other support cells that surround the developing fetus. Adult stem cells, on the other hand, are usually what we call multipotent, and these are cells that can generate several different types of tissues. Classical examples are that of the hematopoietic system, which can generate the blood cells and the immune cells of our body, and then, in addition, hair follicle stem cells, which we work on in the laboratory, and these, as I said, generate the epidermis, the sebaceous gland, and other hair follicles. Sometimes stem cells of the adult can be what we call unipotent. Classical examples of that are the germ cells, male germ cells generating sperm and female germ cells generating oocytes. But then in addition, there are specific stem cells that reside within the epidermis that apparently only give rise to the epidermis, and those that reside within the liver that give rise to the hepatocytes of the liver. So if we start at very early stages of development, in fact, for the human, we can start at the stage of oocyte and sperm to generate a diploid cell. As soon as that fertilization process is done, now the cell can begin to divide. And when it first starts to divide, it can get all of the way to the 16 to 30 cell stage as cells that all are basically the same. And these are totipotent cells. These have the capacity to generate not only the fetus, but also all of the support cells of the fetus. And then as development proceeds, the inside and the outside of the embryo begin to be distinguished from one another. A process known as compaction occurs, and now up to about the 50 to 100 cell stage, the embryo develops and it forms an outer layer of trophectoderm cells which provide the support for the developing fetus, and an inner group of cells known as the inner cell mass, and these are the cells that will basically produce the fetus. And now in additional stages of development, the tissues and then eventually the organs of the body begin to develop. And now in the adult, there are stem cells that progressively become more restricted in their options that then reside in these various different adult tissues. And in fact, we're learning that most tissues of the body, in fact, if not all tissues of the body, do have some type of cells that can undergo regeneration, at least under a limited series of events. So where do embryonic stem cells come from? They come from the blastocyst stage of the embryo, and in the human, as I mentioned, we're about 50 to 100 cell stage, and they come from the inner cell mass cells. And this entire progress of development up to the 50 to 100 cell stage can take place in a petri dish in the laboratory one doesn't need a womb for this process, and one can produce the inner cell mass cells and the trophectoderm cells, the support cells. And now if you take these inner cell mass cells and you culture these cells, that's where embryonic stem cells come from. And as soon as one cultures these cells, then under the appropriate conditions, effectively those cells can continue to divide and do so endlessly. And this is basically where these cells are derived from. And these cells then are pluripotent and can differentiate into a variety of different tissues. So I think there's no one in the audience who is not familiar, at least to the extent of knowing the promise, that there is promise of embryonic stem cells for regenerative medicine. And the reason now is quite clear, that basically with the ability to generate virtually 220 different cell types in the body, then the capacity to generate a variety of different cells to cure or to at least treat or improve the conditions for a number of different genetic disorders for which we currently have no other cures. And so clearly with this type of research, there also is going to be ethical issues involved. Why is there so much controversy? I'll try to present very briefly both sides of the coin. And with regards to one point, a frequently heard argument is that human embryonic stem cells are typically cultured from fertilized eggs, and this involves the destruction of a fetus. The counterpoint to that is that human blastocysts and embryonic stem cells can be created with an egg and a sperm in culture. There's no womb required, and that this involves the same technology as that which is already in place in vitro fertilization. And in vitro fertilization received quite a few years ago now, quite the same types of concerns. And now that process is in practice. And I think many of you in the audience are probably familiar with someone who has had a child by in vitro fertilization. So it's becoming more and more common over time. And with the use of in vitro fertilization, that also results in a lot of unused embryos after a successful in vitro fertilization has taken place. There are often many spared embryos, and they're either discarded. They used to be discarded generally until people began to be concerned about that, and now generally they're kept frozen. But it's actually becoming a problem in terms of what to do with these unused embryos. And so on the one hand, you could argue, well, we could use these embryos for someone else who would like to have a child, or the counterpoint to that is that we could use those for research to generate embryonic stem cells, and then to be able to differentiate these cells and begin to make tissues to treat various disorders and injuries for which we have no cures. Then you get down to another layer of opinions and beliefs, and these become harder and harder to argue and provide points and counterpoints to. One of the classical arguments that one deals with is that this is creating a slippery slope to reproductive cloning, and that this can fundamentally devalue human life. You also have heard the arguments, I'm sure, that a human embryo is human life and therefore entitled to protection. And yet you can counter that by saying, well, societies make laws, they can and should legislate what will and will not be allowed, and therefore we can draw the line with regards to a slippery slope and engineer or put into place the appropriate laws to separate the two. And another argument that you can make is that nowadays the unfertilized oocyte is all that's required, and these cells are haploid. They cannot generate human life on their own, and therefore the issue of human life is not a valid one. In fact, every reproductive every female of reproductive age generates oocytes that are generally unutilized every month, and so you could make these kinds of arguments to use those for regenerative medicine purposes. So what are the potential ways to alleviate the ethical concerns, given that with religious issues, those may never be circumvented? And so one of the ways in which scientists have developed over the last 10 years or so the technology is basically the technology to take human embryos at the 8-cell stage, remove one of those cells, use those cells for something else, such as embryonic stem cell technology, and the 7-cell embryo can still sense that there's a cell missing and generate that 8th cell without generating a human being that's going to be missing an arm or a leg. And so basically there can be a normal fetus produced from the 7-cell embryo, and the 8th cell could still be used for some important purpose. So there would not be destruction of human life in that process. In fact, this technology was originally engineered for the treatment or for in vitro fertilization for families that had a very high risk of passing on a very severe, sometimes life-threatening autosomal dominant trait to their offspring. And for many of those couples, the election was not to have children as opposed to placing one of their children at risk. And as the human genome became apparent and sequenced, geneticists now use this type of technology in order to be able to identify which embryos are healthy and which embryos carry the particular defective genetic trait. But the technology is not without risk to the embryo, and for some it's still not going to be ethically acceptable. So where does the world currently stand on the ethical issues? Well, countries such as the United States, Austria, Italy, Ireland, Germany believe that it is ethically wrong to conduct research on human eggs and sperm because there's destruction of human life. And when I'm mentioning countries, I'm mentioning governments of the countries, not necessarily what the people believe. With regards to governments, Singapore, United Kingdom, Israel, Sweden, Spain, Belgium, and more recently France, now take the stance that in fact it's ethically wrong not to conduct the research on embryonic stem cells when it has the potential to save human lives. And so I make the point because there's really no moral high ground in this set of ethical controversies. There's really different ethical arguments that are in many ways equally valid. It really depends upon really where one falls along the lines of religious beliefs as opposed to scientific arguments or opposed to who has the ethical moral high ground. And I point out that France currently permits research on existing embryonic stem cell lines and IVF embryos that would otherwise be discarded. So now if we go into the biology, what are the biological hurdles if we set the ethical hurdles aside? And the major biological hurdle that existed about 10 years ago, maybe a little bit more than 10 years ago, that seemed insurmountable at the time was the fact that if you generate embryonic stem cells from someone else and now use those to generate neurons or pancreatic islet cells that could be used in a patient to treat a particular disease, that basically the immune system recognizes these cells as being foreign and is going to eliminate them. And so here is the great capacity of our immune system to recognize cells as being foreign now being turned against this type of technology. And so circumventing the immune system at the time seemed like a relatively large hurdle until the use of nuclear transfer research came onto the scene. It's often mistakenly referred to as human cloning. It's the technology that was used to create Dolly, the cloned sheep, and scientists in society are opposed to human cloning, and so what is this technology and how can it be used in an ethical manner? And so basically the overall methods for nuclear transfer are very simple ones. Starting with an unfertilized oocyte, which is haploid, one merely removes the nucleus from the oocyte and discards that nucleus. And then you can replace that with a nucleus from a diploid somatic cell, an adult cell. And my laboratory has used the adult skin stem cells, which we purified several years ago. And we took the nucleus of the adult skin stem cell and introduced that into the, in this case, mouse oocyte, using mouse as our model system for study to create the diploid cells. And once you've created this diploid cell, you can now culture that to the blastocyst stage and implant that into a host mom. And these are some of the cloned mice that basically we generated in collaboration with Peter Mombarts and his co-workers at the Rockefeller University. And we did these studies. They were published earlier this year. And I just show you a couple of movies. On the first one, here you can see one of the mice that was initially cloned from skin stem cells in the laboratory. It's perfectly healthy. These mice can give rise then to litters, and the litters as well in these two cloned mice are basically perfectly healthy as well. And so the frequency at which this happens is not great. It's about 5%. But the fact that these mice can be generated and create all of the proper 220 different cell types of the body and put them together in the right form and fashion really says that, that the adult somatic cell does have that potential, and that the oocyte cytoplasm has somehow the potential to take this more restricted cell, a cell that can normally only differentiate into sebaceous glands, hair follicles, and epidermal cells, and now end up being entirely reprogrammed, at least reprogrammed sufficiently to be able to generate all of these 220 different cell types of the body. And so I think that's quite remarkable. And on the other side, another aspect that's occurs at even higher frequency is the value of this type of technology for human research. Because in fact, if we now go to human instead of mouse, obviously we can't do cloning, but we can go through these same first steps. And now if one simply takes these hybrid cells, cultures them to the blastocyst stage, and lets these blastocysts hatch, one can effectively then generate embryonic stem cells. And now these embryonic stem cells would be containing DNA that is basically matched to the patient that had the skin. And since it's very easy just to take a skin biopsy and culture these skin cells out of the biopsy and basically put a Band-Aid on the skin, that heals very quickly. And now one can basically alleviate the immune system problems and generate embryonic stem cells tailored to a particular patient. I point out that this technology, as of yet, has not been successful in humans, but I will touch upon this later. In fact, it's just been in the last couple of months where scientists have now reported the successful generation of embryonic stem cells from macaque monkeys using nuclear transfer research. And so I think most of the people in the field of stem cell research believe that within 2008, people will be successful at culturing human embryonic stem cells from nuclear transfer. As of now, I believe that France still does not allow that technology. Many countries of the world draw the line between generating embryonic stem cells from IVF versus generating embryonic stem cells from nuclear transfer because of the slippery slope issues, and that this technology could be used for human cloning as well. And again, I would argue that one might, as soon as this technology is in place, my guess is that people will draw the rules, make very strict regulations against it, because this type of technology really gets one around the issues of immune rejection that otherwise are going to be problematic. So at the moment, most people are using this type of technology for research purposes. And then with regards to the research, what is actually happening to that nucleus when it's exposed to oocyte cytoplasm? Well, this deals with the overall research of epigenetics. Basically, every cell in the body has the same or very similar genetic constituency, and as development proceeds, cells become more and more restricted in their options and their ways and the genes that they express, and they do so by virtue of getting methylation and modification marks to their DNA, as well as to the proteins that associate with the DNA, the histones and transcription factors, etc. And all of these marks that are transient marks, they're reversible marks, are basically epigenetic marks. So the epigenetics, the methylation modifications, histone acetylation modifications, have to be erased, and somehow that oocyte cytoplasm has the capacity to do so. Whereas all of these different cell types have the same genetic constituency, but they're expressing different sets of genes due to epigenetic modifications that take place in development. So what is it about the oocyte cytoplasm that affords this potential for reversing these epigenetic marks? I think is a really interesting question, and one that a number of laboratories have started to focus on. So once one gets embryonic stem cells, now the challenge is, how do you differentiate these cells? If you were just to take embryonic stem cells and inject them into the body, you'd generate a tumor, a teratoma tumor, that would basically be a tumor comprised of many different cell types of the body, skin cells, muscle cells, tooth cells, blood island cells, really hideous tumors. And obviously, one has to be able to then differentiate these cells in culture, make sure that there's no spare undifferentiated embryonic stem cell, and get a pure population of the particular cell type of interest. And here, research has really made a lot of strides. There are very few cases now where people have not been successful at getting very pure populations of different types of cells, simply by exposing embryonic stem cells to the particular growth factors and hormones and the like culture conditions that are necessary to optimize the differentiation of these different types of cells. And every month there are articles published where someone has succeeded again in generating a different type, a new type of cell from embryonic stem cell differentiation. So this just shows you one example of differentiation of muscle from embryonic stem cells, here marked in green, and you can see that the muscle cells then can operate in sync. And I show you one example coming from Johns Hopkins University of a rat that has been paralyzed, and what happens when you take embryonic stem cells, in this case human embryonic stem cells, differentiate those into neurons, and then inject those into an immunocompromised or immunosuppressed rat. And on the left-hand side, you can see the rat initially before treatment, and the rat has clearly got a severe spinal cord injury and doesn't have any use of its back legs. And now if you look 6 months later after being treated with these human neurons that have been differentiated from embryonic stem cells, now there is not perfect movement, as you can see, but on the other hand, there's definitely a use of these animals' limbs, and you can even see some evidence of coordinated movement of these hind limbs. And so I think this is really what has captured the imagination of researchers with regards to this type of technology, and starts to illustrate the potential of this type of technology for the future. So nuclear transfer-derived then embryonic stem cells have the potential for regenerative medicine, but I point out one additional aspect, and that is that an area I think of as great an interest to the research scientists and to the pharmaceutical companies is that because it's now possible to use nuclear transfer research to generate or tailor-make embryonic stem cells to particular patients, one can begin to think about late-onset diseases, perhaps slowly developing neuronal degenerative diseases, and being able to take a family that has a very high frequency of passing along a particular genetic disorder like that, and taking the kids at age 10, culturing and making embryonic stem cells, and now having the capacity to generate neurons from those embryonic stem cells. And now one has finally material that one can utilize to begin to look at the slow progression of degeneration of a particular cell type, for which right now there's really no available material. And if one can then have that capacity to do so, pharmaceutical companies are extremely interested in this, because it really then begins to afford the option of being able to then provide screens for these cells to look at what types of chemicals or small molecules might be used in order to be able to slow the progression of a particular degenerative process. And so all of these types of research and pharmaceutical applications now become open with regards to nuclear transfer research. And yet at the same time, for some, and for some countries, the unfertilized oocyte is still required for this type of technology. And there's still going to be religious concerns, and I'm not sure that Italy is going to be able to convince the Pope that this is really something to move forward with. So what can be done under these circumstances? One can then move to the issue of, could we take adult somatic cells and somehow know enough about that somatic cell to revert it back to an embryonic stem cell-like state? And initially, I think many of us were skeptical that this could even be done. It seemed that the oocyte cytoplasm was capable of doing that, so obviously there's some way in which it can be done, but it seemed like such a complex problem that it wouldn't have a simple solution. And yet, within only the past year, Yamanaka and his co-workers in Japan succeeded in taking adult skin cells and basically introducing 4 different transcription factors, KLF4, OCT4, SOX2, and c-MYC, using retroviral gene transfer into the adult skin cells and converting these cells into an embryonic stem cell-like state. They refer to these cells as induced pluripotent stem cells, or IPS cells, and obviously these cells now can differentiate into many different cell types, and yet they only have these 4 genes now introduced back into the adult skin cell. So the adult skin cell in its undifferentiated state is not expressing these genes, and these genes now appear to be the genes that are necessary to turn back on the reversion process to reverse all those epigenetic marks, at least the key epigenetic marks capable of now producing embryonic stem cells. So during the conversion then of a somatic to an IPS cell, the endogenous genes turn out to be turned on. And so when you introduce originally the retroviral genes, the endogenous KLF4, OCT4, SOX2, and MYC genes end up being switched back on again. And so really you only need the retrovirus transiently in order to convert these cells. Then the cell's own system now starts to kick in, and these 4 genes now start to be turned on from the endogenous loci. And The retroviral genes turn out to be turned off, again, taking advantage of the fact that cells can recognize normal genes from foreign genes, and basically they turn off the retrovirally induced genes, and what you're left with are actually, if you look later on down the road at many passages of these cells, what you're left with are the endogenous genes on, and the retroviral genes are suppressed in the genome. And so transient expression then is what's required. Are these embryonic stem cell-like cells, these iPS cells, pluripotent? Well, in the case that Yamanaka initially did, and Rudy Yanish did similar experiments at MIT, they could use mouse cells, and in using the mouse cells, they could use the gold standard and ask, can we get mouse embryos and mice? And the answer was yes. The problem was that virtually all of those mice developed tumors over time, and they developed tumors because c-Myc is one of those 4 transcription factors, and c-Myc is an oncogene. And it turned out that in those tumors, the retroviral gene, c-Myc gene, which had been shut off in passaging the cells in vitro, now spontaneously or sporadically turned back on again, and that led to tumorigenesis. So in vitro, however, these cells could be differentiated into neurons, blood cells, vascular cells, muscle cells. Many different cell types had many of the different cell types that were generated by taking normal mouse embryonic stem cells and differentiating them, subjecting them to the various different regimens. So genetic programming then, which is what this is, genetic reprogramming, is to use foreign genes in order to be able to, or foreign mechanisms, genetic mechanisms, to genetically alter the nucleus in order to be able to convert these cells back to their embryonic state, and that differs from the epigenetic programming, which I talked about with regards to nuclear transfer research. So the problem with that for regenerative medicine is that, in fact, in those initial papers, there were 60 retroviruses that were integrated into the genome. About 15 copies of each gene were necessary in order to convert these cells. And obviously, with 60 integrated retroviruses into the genome, this would not be suitable for regenerative medicine. In addition, one of those genes was Myc, as I mentioned, and so obviously that also posed an additional problem. So it would be nicer to be able to avoid genetic reprogramming, or at least to be able to avoid some of these hurdles, which are right now clinical hurdles to the application of this research to regenerative medicine. And so where are we in between? We can do nuclear transfer research, but that requires an oocyte, or we could start with adult cells, and that requires foreign genes being introduced. On the one hand, it's genetic reprogramming. That's not going to be useful straightforwardly for human regenerative medicine purposes. Nuclear transfer might, but it's not without its ethical issues. And so what next? What have scientists done next to try to basically get around these types of problems? Well, I mentioned that with regards to nuclear transfer research, I think the point at which researchers have now come with regards to all of the various different hurdles, getting around the immune system hurdles, starting that research initially with mice, and in fact mouse embryonic stem cells were first generated, I think, in the late 40s, early 50s. This work has been going on for many years. Human embryonic stem cell research, the interest in making human embryonic stem cells is a relatively recent technology, and yet in the end of 2007, just a couple of months ago, we've now gotten, or the field has now gotten to the stage where monkey ES cells can now be generated. And so I think this is really right around the corner in terms of getting to the point where it will be realistic for scientists and clinicians to think about nuclear transfer research in human regenerative medicine. With regards to the cloning of iPS cells, in 2000 and late 2006, early 2007, people were beginning to meet with success at making iPS cells from mouse. And now in 2007, the last couple of months, Yamanaka's group again has now succeeded, along with Jim Thompson's group at University of Wisconsin, in generating human iPS cells from adult skin using retroviral gene infection with SOX4, KLF4, MYC, and OCT4. Now you'd say, well, there are some problems with that, though. What about retroviruses integrating into the genome? Well, adenoviruses differ distinctly from retroviruses in that adenoviruses do not integrate into the genome. And so if one is thinking about this technology with the realization that all you need is transient expression of these foreign genes to be able to convert these cells to iPS cells, and then once that happens, the endogenous genes are induced, now you could say, well, perhaps then it's possible to use adenoviral delivery, because you can passage these cells endlessly once you've generated iPS cells, and as those cells divide, then adenoviruses are subsequently diluted out. So normally for a scientist, the disadvantage with the use of adenoviruses is that it's transient, because as you divide the cells, the adenovirus gets diluted out of the cells. But in this case, it actually works to one's advantage of eventually being able to rid the cell of the adenovirus, and yet starting with the ability of taking an adult cell and converting it. Obviously, there's always going to be that concern of whether or not there is a stray virus there. There will always be the concern of whether or not there was some genetic alteration where pieces of DNA, in particular c-MYC, end up being integrated into the genome. So adenovirus does not integrate. It's lost as cells divide, and this might be a good thing. And you could say, well, what are those factors then in the oocyte cytoplasm? Can we figure out that? Now we have some clues. We know that the oocyte cytoplasm is probably turning on only a very small number of genes in order to be able to revert the cells back to an embryonic state. And I think no one really predicted that it would be so simple as just a few genes. In fact, one thought you'd really need to be able to erase epigenetic marks for lots of different genes in order to be able to convert these cells back to their embryonic state. So now comes the issue of what's so special about those 4 genes, or are those 4 genes even special? Well, again, we're dealing with 2007 discoveries and many recent 2007 discoveries, just the last month or so. But now Yamanaka's group has taken this even one step further and said, actually, we can get rid of MYC. We don't need MYC as one of our 4 retroviral genes in order to be able to get iPS cells. So Yamanaka's group has succeeded in cloning mouse embryonic stem cells just using SOX4, KLF4, and OCT4, and eliminating the need for c-MYC. And the frequency at which they generate iPS cells is not nearly as great, but the fact that they can isolate iPS cells without having the use of MYC is indeed a major advance, particularly with regards to now the subsequent experiments, which I'm sure the laboratory is doing, and that is taking it to the level of, do we get healthy mice and mice that don't develop tumors over time? Well, now you get to KLF4 and say, what about KLF4? My laboratory was particularly interested in KLF4. In fact, we removed KLF4 from the mouse genome back in 1999 when we reported this. We did so because KLF4 was a gene that was involved in late-stage epidermal differentiation. It seems ironic that here's a gene involved in late-stage epidermal differentiation, and yet it's required for embryonic stem cells. And in fact, it's not required for embryonic stem cells. We first generated embryonic stem cells from mouse in order to make the mice that lacked KLF4 in their genome. So it's been known, at least since 1999, that KLF4 really isn't needed. And in fact, now, George Daly's group, just in the last month or so, using, in this case, human iPS cells, has now said, actually, you can eliminate KLF4 as well. But it turns out, and he also showed that you can eliminate mix, so that takes Yamanaka's study on mouse one step further to human. And now it turns out, well, can we eliminate both of those? And what are they doing? It turns out you can't eliminate both of them. You can eliminate one or the other, so maybe they're doing something similar. And then what is it that SOX2 and OCT4 do? Because those are the genes that seem to be absolutely essential. And OCT4 is only present in embryonic stem cells. It's not present in any adult stem cells. SOX2 is present in a few. So scientists have known for quite some time now that at least one gene that appears to be critical in, as a downstream target from SOX2 and OCT4 is NANOG. This too is another transcription factor. And when KLF4 or c-MYC is missing, NANOG seems to help by restricting differentiation options. So as long as you can control the expression of NANOG in the embryonic stem cell, then the embryonic stem cell appears to be held in terms of what its differentiation options are, and it will be maintained in an undifferentiated state. And then as you subsequently start to lose expression of NANOG, you start to be able to open up the options to the embryonic stem cell to differentiate. But NANOG has been shown to be absolutely essential for generating germ cells, the male and the female germ cells. So NANOG is an essential gene. You can't get very far without germ cells. And yet NANOG is dispensable to generate embryonic stem cells. So here are two transcription factors that are essential, and for a long time people thought they were essential in generating the expression of NANOG, but in fact it turns out that NANOG is not necessary in embryonic stem cells. And this was work done by Austin Smith at the UK and Cambridge and published again just a few months ago. So NANOG null mouse embryonic stem cells can be maintained in culture, and he's also done injections to make teratomas in nude mice or immunocompromised mice and shown that NANOG can be eliminated, and you still have the capacity to generate all of these different cell types in a nude mouse. So what is NANOG doing then? NANOG appears to be acting in many ways like a rheostat, which then finally adjusts the options that are afforded to the embryonic stem cell. The more the NANOG, the more the embryonic stem cell-like, the less differentiated the cell is, and the fewer the number of cell options are as long as NANOG is maintained. And so the expression of NANOG appears to be important in terms of its regulation, but it also appears to be really acting as a rheostat, not necessarily essential, but one that is clearly one of the important genes that has an impact on maintaining an undifferentiated state. So now I'd like to just get to the very last aspect of the biology dealing with what's necessary to maintain an undifferentiated state. And this is really research that comes from Rick Young's laboratory at MIT, where Rick has used technology known as chip-on-chip technology, which allows him then to look at what are all of the different genes in this case, the human embryonic stem cell that bind OCT4 and that bind SOX2. Maybe it's not just nanog that is being regulated in some way. In fact, it turns out that nanog is being regulated in an interesting way. But what are the other genes that OCT2 and that OCT4 and SOX2 are regulating? So he used chip-on-chip technology, and what he's discovered over the last couple of years is that SOX2 and OCT4 bind to the same genes, and in fact they also bind with a group of other chromatin-modifying genes called polycomb genes. This is a repressor complex, and it appears that in many cases in the human embryonic stem cells, that OCT4, SOX2, and polycomb proteins are acting together as a repressive complex to repress genes, as opposed to be involved in activating the genes. What types of genes do they typically repress? They typically repress differentiation genes. And so when you begin to look at the whole scale genome of what SOX2 and OCT4 are doing, we're now starting to get a picture of just how the undifferentiated state is maintained, and then how the loss of or changes that occur in this complex then ultimately lead to additional epigenetic changes that then allow the relief of repression and allow these genes to be expressed. So as development proceeds, then the differentiation options become more restricted, but cells start to express particular clusters of differentiation genes that in the embryonic stem cell are being repressed by this group of proteins. So hopefully now you're starting to get a view of just what the issues are, both at a clinical level, at an ethical level, and also at a biological level. And at this point, I'd like to just finish today's lecture with a little bit of a discussion about tapping the potential for adult stem cells. I've focused the lecture entirely on embryonic stem cells to this point. Hopefully that reason is clear. But I'd like to not discount the importance of adult stem cells, particularly because those are the cells that I happen to work on. And there are reasons for working on adult stem cells as well. So adult stem cells, if you recall, are the resident cells of a tissue that can repair the damaged tissue from injury or aging. And some tissues have many more stem cells than others and have the need to replenish their tissue or their cells of their tissue constantly. And a perfect example of that, of course, is the epidermis and the hair follicles, where every 4 weeks' time you've got a brand new epidermis on your surface of your skin. Your skin's always being subjected to all sorts of physical and chemical traumas of the environment, and it's important then to continually replenish the surface of our skin, and that happens throughout life, which is really one of the reasons why my laboratory has been so interested in this as a model system for understanding stem cells and their behavior. In addition, hair follicles, if one's lucky, throughout the course of your lifetime undergo periods not only of rest but also periods of growth. And so there's this continually replenishment of these cell types in the body. The hematopoietic stem cells are another good example of that, where on a daily basis there are thousands of cells in the blood that are continually being replaced, and they're replaced by a constant source of hematopoietic stem cells being differentiated into these various different tissues. Intestinal stem cells are another example. And interestingly, the examples, when you start to think about it, just for the future lectures that we'll be discussing, the cells that need to turn over rapidly within the body are often cells that are also very much prone to human cancers. And there's certainly a link, and I'll go through the biology of what those links are. So other tissues have very few stem cells, and in fact, it's really only been in the last decade that we've even thought that the nervous system was not simply that neurons were not simply post-mitotic, but actually there are neural stem cells. And in fact, just in the last couple of years, not only is there one neural stem cell compartment, but there are many neural stem cell compartments in the body. Pancreatic tissue, Doug Melton's group has shown that that the only type of cell that exists that can produce a pancreatic islet cell in the adult is a pancreatic islet cell. These cells do have the capacity to divide. On injury, they will divide, and so in that sense, you can call them a unipotent stem cell. Doug Melton would say, well, there's not really a stem cell. It depends on if your definition is to allow a unipotent cell being called a stem cell or a progenitor cell, and just how long-lived it is. But pancreatic tissue can regenerate to some extent, but it's very limited. So why even think about using adult stem cells for purposes of regenerative medicine? Well, if you're only interested, if you've got a tissue that has a good source of stem cells that's accessible, and you're only interested in using those cells to replace a particular tissue, then you're much better off starting with the adult stem cell than you are with an embryonic stem cell. You avoid all of the risks of having an undifferentiated escaper embryonic stem cell in your differentiating cells that you're now going to apply to a person, and the risks are low for obtaining any kind of unwanted differentiated cell. If all I want is an epidermal stem cell to repair epidermis, then there's absolutely no point in my starting with an embryonic stem cell to do that, other than for an interest in the biology, which is a different question. So in fact, back now 30 years ago, when I was working in Howard Green's lab, a little bit later than 1975, but in 1975, Howard Green began working on human embryonic stem cells, and he learned how to grow these embryonic stem cells last feeder layer, which was actually the type of method that was then used and has been used successfully of growing human embryonic stem cells and mouse embryonic stem cells. This technology was originally used by Howard Green to be able to grow epidermal cells, and if chosen the right atmospheric conditions, the right combination of oxygen and carbon dioxide, and the right body temperature, basically these cells could be propagated, and propagated quite endlessly, several hundred generations in culture. In fact, every now and then when my laboratory now at Rockefeller University wants to study human embryonic stem cells for some reason, I usually like going back to the liquid nitrogen tank to pull out cells that I cultured from human skin back when I was in the laboratory. And so these cells last for an endless period of time, and they can be cultured. And here you can see that the cells can be coaxed to divide quite rapidly in culture. There are several cells, mitotic cells in this view. And in addition, over the years, my laboratory and other laboratories, particularly a Seleno's laboratory in Nice, has devised the mechanisms for being able to recreate human epidermis. In this case, epidermis starting entirely from scratch with single cells in a petri dish, coming initially from a small biopsy of a patient's skin. And in fact, as soon as Howard Green began to develop the technology for culturing human embryonic stem cells in the laboratory, then the realization that these cells in culture could actually be used in a clinical setting for the treatment of burn patients came on the scene. And this still, after 30 years, and now utilized across the world for supplementing the treatment of human burn patients, stands as an example, a very successful example of using stem cells in a clinical setting. So when people argue that there's no examples yet of stem cells being used for regenerative medicine, in fact, we can go back to the late 1970s when this was developed. And here you can see an example, one of the first examples, where this entire region of skin was generated entirely from cells in culture and used in grafting. And in fact, in these cases where there are very badly burned patients, it turns out that you can generate more cells in culture than you can graft on the patient at any one time because of the issues of exposing too much surface area of the skin from the patient to infections. And in addition, the epidermis provides a barrier not only for infections, but also to keep essential bodily liquids in. And so if you didn't have an epidermis, you dehydrate within about 24 hours. And that's really the only reason why it's important to have these artificial skins used. They're really used transiently, pig skin or artificial skin, used transiently as a covering, but culturing human epidermal cells in a laboratory, that's real cells, and you can culture them from an area of the patient's good skin, and again, graft those back onto the patient. So this is one example, and another example successfully used in a clinical setting for now over 3 decades is bone marrow transplant, which is readily acceptable for a variety of different needs where there are problems with hematopoietic stem cell generation, and bone marrow transplants have been successfully used in many different avenues for the regeneration of the immune system in patients. So again, several at least successful examples. No successful examples yet of human embryonic stem cell research, and these are really adult stem cells. So not to discount the importance for adult stem cells, it's just that embryonic stem cells have clearly much broader potential. So I'll finish then by just touching on the on neural stem cells, again because this is such a fascinating area and one that's developing so rapidly. And it wasn't but a decade ago that researchers began to realize that there was an area in the brain that is known as the subventricular zone, and there are a source of neurons that can be cultured, and these neurons can also differentiate into transiently amplifying cells, shown in green here. The blue cells are the stem cells, and then those cells can then so they then produce rapidly dividing progeny, and then those cells produce the red cells shown in A here, which are neuroblasts, and these neuroblasts from the subventricular zone will migrate to the olfactory bulb and form interneurons. And so these are bona fide stem cells. Rusty Gage and his group at the Salk Institute has cultured these cells, and these are just examples of the cells, in this case marked by green, and not to color code them, they're miscolor coded here. They should be color coded by blue fluorescence rather than green fluorescence. But you can see these cells actually can divide quite rapidly in the laboratory, and they divide symmetrically. And then one of my favorite examples of neuronal stem cells has only been published in the last couple of months, and it's an appropriate place to finish because it deals with stem cells that are neural crest derived. And I always feel that with Nicole Douaran from France really being the pioneer of the whole field of neural crest cell biology, that it's an appropriate place to finish by talking about neural crest derived stem cells. They were recently described in a region known as the carotid body, and this is a region where recently researchers have realized that not only are there neural stem cells that are hiding within this body, but in fact, these are the cells that generate dopaminergic neurons, and they can do so in response to hypoxic conditions. And so it's been known for years that animals that are placed in an environment where there's low oxygen condition, or for us as animals, if we're placed at the top of Mount Everest, we're in low oxygen conditions. We can't survive that for an extended period of time, but we can survive it for a while. Why is that? And it turns out that the carotid body starts producing and generating more of these dopaminergic neurons in response to low oxygen conditions. And that allows this transient supply of ventilation to the tissues of the body and allows the tissues to survive for a while under these circumstances. And so this is a fascinating example. I think one of the reasons why biologists are so interested in it is that up until this point, it's really been damage or injury that is a spinal cord injury or the like that has been used in terms of trying to see what would neural stem cells be able to do, placing these cells in culture. And here's a fascinating example of a transient regulatory mechanism to control stem cell proliferation in the nervous system. So on Tuesday's lecture of next week, we'll pick up and talk about the skin in much more depth, and I'll tell you about what my laboratory is doing with regards to understanding skin stem cells, and really from the perspective of how do you make tissues from stem cells? How does that process occur? And once you understand that process, how do you make multiple different types of tissues starting with the same cell? A fascinating question, and one that is a very complex one, and I'll touch upon the research that we've done that starts to elucidate those complexities. And I thought that for all of you in the audience, even though you may not work on skin, and skin is not always necessarily the research paradigm that people think about, that in fact nature clearly has had a lot more fun and fancy in creating body surfaces than she has in creating any of the ugly organs that most researchers use as a model system for their own studies. And I think that even we as human beings have had a fascination with our skin in a way that has prompted us for eons, almost since our existence, different cultures of the world to adorn upon what nature has given us. And so I think there's a fascination about skin in all of us and all of nature, and I'll try to convey that to you next week when I continue the lectures. And I would like to thank again Christine, this time in fluent English, for her very kind invitation to give these lectures. It's such an honor for me to be here, and it's wonderful to be in Paris. It's the magnificent city of the world, and it's my pleasure to be able to have the opportunity to speak with you. Thank you.